So next speaker is Professor Daan Crommelin from Netherlands, from the Utrecht University. Basically, he's known to everybody who ever worked with liposomes and similar systems, and he will talk about induced stability studies, which I know he likes very much, of COVID-19 vaccines.
Okay, thanks, Hedwig. Yeah, yesterday I talked about the stability of mRNA. I mainly talked about the the chemistry and the chemical implications for instability and how to do something about that. Today I will be much more down to earth. I'm a pharmacist by training, and although they never allowed me to work in a pharmacy, which is, I think, quite a good idea, I still have this interest in, well, what are these things doing in real practice? So, induced stability of the COVID-19 vaccines is the story I would like to talk about. Everything that I'm talking about can be found in these publications. They will be available, I guess, later after the conference, so you can pick it up. It tells you the whole thing. I just pick out a couple of new things that are not in there yet, because they came in, this new information came in the last couple of months. This is a slide and a table taken from one of these publications, and I would like to point you to the stability at 2 to 8 degrees centigrade and the stability at room temperature. This is an old slide in the terms of this was the situation December 2020. Oh, that's a long time ago, isn't it? I mean, what were we young at that time? So things have changed a little bit, but not basically. Stability in the refrigerator and stability at room temperature, there were limitations, and the industry, together with the regulators, pointed that out. And they gave these ranges. Very important for the rest of the story is that the inserts and the inserts for how to use these vaccines clearly indicated that whenever you had to mix things, that you had to do that gently turning things around, swirling, not shaking. So these were the instructions, but there were no data. There were no hard data. And it took a while before the first hard data appeared. And this is a story from Italy, pre-drawn syringes on Comirnaty, that's the Pfizer vaccine. Demonstration of stability in real-life conditions. So this was for me. I mean, this answered my question. How are these things doing in real-life situations? So on the left-hand side, the storage conditions, syringe or glass vials, and they looked at the temperature. They used the temperatures that were prescribed, and they also looked at mechanical stress because they had to gently turn them around and not shake. So they looked at, pretty primitive analytical tools, but I mean, what can you expect? Chain integrity, gel electrophoresis, and dynamic light scattering, nanoparticle tracking analysis, and size and shape and TEM. Here you see 2 TEMs, and I hate negative staining TEMs because they don't tell me anything, but okay, they are here. And the outcome of this story was there's the conclusion with this limited set of physicochemical parameters. Those are my words. 5-hour storage at room temperature, okay. Shaking, detrimental. You really get big aggregates. So the gently turning around, swirling was definitely a good precaution. Then a little bit later, a British group did actually sort of the same thing. I don't give you details. The conclusion was the same. As long as you don't shake it, you're okay. So what was it with James Bond? Shaking or swirling? What was it? I forgot all this. Anyway, he doesn't exist anymore, so it's the good old times. So the same story, both in Italy and in Britain, they found that don't shake it. Then there was a long time nothing, and until 2 months ago, I think, this also from Italy, again from Italy, assessing the physical stability and intercellular trafficking of mRNA-based COVID vaccine. I was interested in the first part, the physical chemical stability, because you couldn't find in the literature any data on the sizes of the Moderna vaccines. On top you see the Comirnaty, so the BioNTech Pfizer, DLS structure, and again this beautiful negative staining picture. And then interestingly, the Moderna DLS showed that that was not a single distribution, but there was a hump there. See, at higher diameters was a hump. This is relative intensity, so it's biased to the larger size range, but it's very clear that the Moderna vaccine, when it was used or when it was introduced, was not completely unimodal. I found that interesting because I didn't expect that. But I mean, this was the way it is. As long as you don't shake it, it stays like that, and it apparently worked in vivo. So I looked at this paper in more detail, and this group looked at the vaccine stored at 60 degrees. Particle size, again, the Z average, so the intensity-based diameters, unstressed 8688. So you see, indeed, the diameter, the average diameter of this Moderna product was quite a bit higher than the Pfizer product, the BioNTech product. After 4 days, nothing happened, and after 8 days, nothing happened. So basically, you're okay, and this is at 60 degrees, so it's pretty high. The polydispersity index didn't change. So in that sense, the indications that were given in the table that I showed you before, no problem with that. This group also did something else. They looked at the stability in the syringe and the interaction with silicone oil. And as a pharmacist, I know that you always have this problem with silicone oil in injections and all kind of hydrophilic or surfactant type of products, liposomes and colloidal systems in general. You have to be careful with the silicone oil interaction. Here, no issue at all. Unstressed, 10 minutes, 40 minutes, 90 minutes, no changes in particle size, no changes in polydispersity. So, as a pharmacist, as a scientist, I thought, well, that's fine. That's not too bad. Why weren't we not aware of these data? Why did the industry not publish this? Because a lot of people were very suspicious about this whole thing. So be open and candid about it. So this is in the end what happened. No problems, fine. And now I turn to and now for something completely different. And I turn to Denmark. Hamlet, to be and not to be, or there's something rotten in Denmark, as Hamlet says. Interesting, I'll leave you with this, because this is not really in situ, this is real in practice performance of these vaccines. This was published in a peer-reviewed paper by the 3 people over there, Batch-Dependent Safety of BioNTech COVID vaccine. Who saw this paper? Who saw the data? 1. Nobody else? 2, 3. Very good, very good. Regulators should see. Suspected adverse effects on the y-axis. So alarming. And here the number of vaccine doses per batch. So at the bottom you see these are huge batches, and these are very small batches. You see the small batches, a lot of trouble. Big batches, no trouble at all. How is that possible? Was Pfizer not able to produce these things properly in the beginning? Was it a sort of, the authors used the term Russian roulette? Or is there something else? Well, this is where we as a delivery scientist sort of meet reality. So we have to go to the epidemiologist. And that also meant that as soon as this was published, there were a number of people who wrote an article about this and told the world that it was all nonsense. That you had to really look at it with a completely different set of eyes. You have to put on a different glasses. But it is still there. And I haven't seen anything from industry to sort of explain what happens here. And I think here again, I've sort of also followed this conspiracy theory type of things. You really have to step up and explain these things. And look at these epidemiologists. They already did that. And I really recommend you to read both articles. They are first-class examples of what you should teach your students in the first and second years of their training as a pharmacist. How you can get these data there and how biased they may be. Thank you.